gotta make a change. It's time for us as a people to start making some changes. Let's change the way we eat, let's change the way we live, and let's change the way we treat each other. You see, the old way wasn't working, so it's on us to do what we gotta do to survive. And still I see no changes. Can a brother get a little peace? It's war on the streets and a war in the Middle East. Instead of war on poverty, they got a war on drugs and the police.
Alright, today we're still in SB2. Today we're going to look at describing the relationships between changes in DNA and the potential appearance of new traits, and examine the use of technology in the fields of forensics, medicine, and agriculture. Mutations. Mutations are changes in the DNA sequence that affect the genetic information. These changes in the sequence are then able to be passed on to successive generations. Two kinds of mutations are gene mutations and chromosomal mutations. Gene mutations. You got point mutations, which affect only a single nucleotide. So one kind of those is substitutions, where you substitute one nucleotide for another. So over here we have the DNA sequence GCA, which codes for mRNA, to be CGU, which codes for a specific amino acid. If that C is substituted with a T, then that's going to code for the mRNA CAU, and which is going to code for a completely different amino acid, which may code for a different protein and a different trait. And then you have frame shift mutations, where those the three nucleotides that make up the codon, if you insert or delete one of those, then that's going to cause that frame to shift over either to the left or to the right, and causing it to code for a different amino acid and a different protein as well. Chromosomal mutations. So those were gene mutations, and they were just specific points on the chromosome. This is when the entire chromosome is affected. You can have a deletion where the B gene of this chromosome is just completely deleted. You can have a duplication where the B gene is duplicated and you have two, where it's A, B, B, C. An inversion is when on the same homologous chromosomes you have parts that are reversed. So we have A, E, D, C, B, F instead of A, B, C, D, E, F. And translocation is when it happens between two non-homologous chromosomes and results in the two chromosomes being different. Applications to genetic engineering. So humans have learned how to manipulate the DNA, and some of the simpler ways for one is selective breeding. That's when humans take the animals or plants or whatever that they want, that have the traits that they want, and only allow those to breed. You have hybridization, which is crossing dissimilar organisms to produce a variety and trying to get the best traits from each different organism. You have inbreeding, which is almost the opposite, when you're crossing similar organisms to maintain specific traits, which happens in dogs a lot. And you can also increase variation by causing mutations by adding chemicals or radiation. Yeah, erased. Technology in forensics, medicine, and agriculture, and you've already done a project on this. So you can take the DNA out of a cell, DNA extraction, then you can cut it and look at different sequences at a time, and then you can separate it out through a process called gel electrophoresis and so these allow, like, detectives to take DNA to look for specific individuals that they can compare it, and in medicine and agriculture, we have things like transgenic organisms, which is when you take a trait from one organism, a gene from one organism, and put it into a completely different organism. Like they taken the gene for glowing from a firefly and put it in a tobacco plant, and you have glow-in-the-dark tobacco for some reason. Or cloning, which is taking a cell from an adult and making a completely new organism, and obviously that organism is going to be exactly the same. Also with this one, transgenic, they can take human genes and human DNA and put it in things like mice to test medicines on to see how humans would react to the medication. And then there's also things like genetically modified food and all these other ways that people are using these in these three fields.